to talk to you about a method that we developed during part of the session. Essentially, it is a metric that will look at gene set and adjust the functional coherence in a global way. Some of the motivation is that there are really in different types of now that produce through these data often are a gene set. So some sort of a list yesterday people were microscopy, and so this should end up with this result. And what we want to do is want to large groups to figure out which ones are important for. In this study, we have devised these metrics based on the genes, gene ontology, and annotations, functional coherence evaluation. So let me start with an example. Say that we've just run a microarray, and with 6 genes here, 7 genes, and everything looks better as a graph, so I put them up there as a graph. We don't know which cluster to study, so what we'll do normally what they're going to do is they'll go back to the Gene Ontology, which is standard for at least for mouse and human. And what they're going to do is they're going to look at the ontology annotation of genes. They're going to try to figure out in some way are any of these annotations really rich. And there are 2 methods here that I have that both do this. One uses the hypergeometric method, another uses the so we're asking a little bit different question. As individual annotations enriched, what we want to know is all of the genes that are associated with these genes in a given cluster, are they all functional? And you might be able to ask that same question using an enrichment-based method, then maybe test, but then you run into difficulties of testing scenarios and so on. So we took a step back and developed a sort of graph-based method to go about this. When I talk about functional coherence, there are 2 main aspects to functional coherence. One is the that are shared genes within a given cluster. So maybe the genes in these here have apoptosis, and if you see that enough times, then you can say, well, it's so that aspect. The second aspect is maybe apoptosis regulation of apoptosis. Well, these genes, these annotations are similar, and we want to have a method that really can take that into account in some way. An overview of the overall method is fairly. So what we do is we use the Gene Ontology, which is represented as a graph. This is the Gene Ontology that you see here in the upper left. So start set of genes, call them genes of interest, GOI, can be different really any experiment. We have microarray up here, but from an experiment from whatever it is. The method was tailored for an arbitrary gene set that is really independent of how you produce your gene set. What we do from this overall structure, we want to find a structure that's small, and it represents all of the that are associated with those genes, and it also conveys the feature. So we essentially a subgraph of some sort, and for this we use Steiner, and I'll talk about the Steiner tree method here a little bit more in a minute. And we also want to describe, so our method involves describe the tree in a quantitative way, using topology, and we wanted to measure to describe this tree. We would model, and then we can do a discriminative analysis and end up with a p-value. Take that back, just the gene ontology again. All the terms have relationships, and it makes it very easy graph itself. Have a Go graph that of ontology. And that's, but one thing that you want to weight is you want there to be a value from common ways to do use based on information out there, and very high method. We, for the purpose of this study, we use. So in brief, essentially the negative probability of a term. You look at the differences of the negative log. These probabilities are directly associated with the number of that are in the ontology for a given term. So we have the gene. We know construction. Let's look at it from a little bit. You can see number of terms that are associated with each one of the species. You can also see, so this the gene is, many of you know, it's three aspects: biological process, molecular component, and we used for the purposes of this biological process method to apply to all three. And the number of genes that are associated with each of the different ends. Another thing that I wanted to say is the ISCB theory. Graph theory is basically just the number of the node. So look at for example, the yeast. If you look at term only, which is just the term degree, that there are on average 3 edges or 3 term. If you were to add the proteins into that graph, numbers up, and you can see the same thing happen the other two organisms. So what one thing we can interpret from is the very large number of and so a more common way to describe a network is to use what's called cumulative degree distribution. If we're going to have our metric is based on degree, if we're going to have a metric based on degree, we want to make sure that the network that we're doing here are somewhat similar. So we're going to use a to look at these just make they're similar. And what we see here, so in the cumulative degree distribution, so the probability degree here, and you see that for the first layer, which are just term-only graphs for the 3 species, you see them in the degree distributions as represented. And for the next 3 graphs, these are the term-only graphs with the term graphs with the protein, and you can see that we still have a lot of similarity. We might have a difference in the numbers or the proteins, but we do have a lot of similarity in the general graphs. So to be able to use this is the second point up here is just to say that a degree-based metric is reasonable between these 3 organisms. And the last 3 panels show the distance. So we used information content again, and these are the information content distances associated with those graphs. You're starting off with this. We have a gene ontology graph. It really is a summary of the terms of the graph. It's in terms of the gene ontology graph. So these are examples of 3 different subgraphs of the gene ontology graph for the genes. So if we have a gene list, 5 genes, this may be 3 different ways that you can summarize, given the gene ontology graph, that data. And so the first one is the old pairwise shortest paths as calculated using this algorithm. What we notice in panel A is that there are a lot of edges to connect these terms, and it's not summary of a gene ontology graph for these 5 genes, summarizing all the terms. Black again, sorry if I didn't mention it first, black squares are themselves, the ovals are the terms, and the shaded oval C terms, those are the actual terms that are being used, and it means the other terms are connecting terms. So if you want to have all the terms, then sometimes we're required to have a number of connecting terms. So if we, instead, all the pairwise S is not necessarily maybe the representation. If we use a Steiner tree in panel B, we have a more concise representation, and a Steiner tree is essentially a tree. So you have a single path that connects any of the two terms, or any of the two nodes in the given set. But you all of this want to minimize the total length, or the total. If we were all just you want a minimization of that. And so that's what we can say representation. The third the third graph here is another Steiner tree of two edges. It may be difficult to see, but there are two edges here. And edges are added because sometimes you have similarity in a gene with annotations that isn't necessarily, and it's not reflected in the semantic similarity. So, for example, cell death and oxidation ontology terms that are used in the gene ontology, it's used to annotate 25 genes. So, a gene-term relationship here that's used very frequently. So what we're going to do is we're trying to reflect this, and they're very distinct. And so what we're doing is we're actually adding new edges here in to reflect this type of similarity. It's a biological similarity, and we also have some empirical data that show that this does help in discrimination for gene sets. And this is how we weight. So essentially, we use style for all the distances in the entire graph, and then counting the number of genes, the more genes that are, the smaller it's going to be. This is for the people who know model the need. Again, we have this Steiner tree. We have a summarizing structure of a Steiner tree. We also want a summarizing structure quantitatively, and there are two obvious ways to do that. One, length C degree, and so if we use length or C degree represent, we'll say that the assumption that it's normally distributed. Use Dario-Watson non-parametric regression to identify here for a given statistic that we can go, and the statistic is going to be a function of n, mean set, and we can use this null model then to get a value. So how well does it work? That's really what we're interested in. We use the KEGG database as a database that specifically works. The KEGG data coherent as pathways, and so how often, if we use all of the pathways in the KEGG coherent gene sets, and are they? Sorry. So these are the data for 3 different metrics. The first metric is the so the terms that are actually used to annotate, and those is just summed over all of the terms. And the combination of the two so the term, we would expect a high degree of terms. So we would expect in a function set. And what we see here, the x's are we can see that the actual values are a little gray, which is all this red line in the Dario-Watson distribution. We see that there's a good degree of separation for seed, kind of we're hoping for. And then for length as well, very small length. So what we see here is that the length, the KEGG are smaller in general than the length for the null. And then the accompanying RC curves here on the right. 3 different species, and yeast certainly does, which is blue. And then other two organisms do fairly well, but they don't perform quite as well as yeast. Put into wheat noise, and they often contain, and especially what we wanted to do here was kind of see how robust they are to noise. So this is a combined metric that I showed in the last, and panel B, C, and D are the mouse and human, respectively. And these, you can see that with fairly degrees of with a fairly low amount of noise, it doesn't actually curve. And then as we get higher, you can see the arrow. So I think that the method is robust amounts of noise. And if we were to just look at and look at the number of significant pathways as a function of these, the ideal trajectory backwards, and we see a little bit that form, and the cerevisiae performs much better than the other two. So we took the method and we applied it to microarray data. So the point of the method, or one of the potential uses of the method, is to be able to filter a large amount of genes. Which ones am I going to go ahead and see? Am I going to on experiments and so on and so forth? So what we're the black are the actual gene clusters that we're keeping, and I'm sorry, the black dots are formed. The blue diamonds are the hypergeometric method. If we were to just use the hypergeometric method for each one of the that are associated, and what we see here is that it's not a function of cluster size. We did not want to say a method has some sort of pattern with respect to cluster as the hypergeometric method. And in the interest of time, so this is the silhouette index is just a measure of how values are, and you can see that changes with another piece of evidence shows that the method is performing well. And really the top is just a p. If you just a p-value, really it's a number, and it really does a lot. And we wanted it does really important to stress is the visualization is extremely important. One thing that does is it allows us to derive insights and that we have made reveal novel hypotheses and so on. Of a cluster that is derived from the RDRD retinal duration model, and what we see here point out is there's a gene which is again the black square with a large number of mutations, and it might be one reason it's interesting to look at. But biologically, this man we really might want to focus more on a region of activity. So this visualization allows us to when genes an area that might be the most. This is the geogenesis for this model. In conclude, just wanted to say that the redistribution is quite similar for using degree-based metrics, and metrics they address both aspects where we have the symmetrics being represented by the length and the seed degree, as I talked about. The method really just low levels of noise, and those are in part when you're looking at the functional coherence of gene sets. Narrowing a little bit, and discover. There were a lot of people are involved in the project, and to thank them. I'd like to thank you.
We have time for questions. You could come to the mics.
Could you check the slide showing the pathway
Yes. Press it.
So anyway, if you can, are you considering all three?
Oh, just the process.
Okay. I mean, there are really two distinct questions I could ask here. One is you should express each pathway to correspond to one process. Right? So should there be any other than one process per KEGG pathway? But then the reason back more one is because actually KEGG pathways are not pathways. The term map is because they actually put more than one biological pathway in each KEGG. So, for example, the KEGG map methionine metabolism has methionine RNA charging and methionine degradation. That's why you're going to have three different Go terms because Go distinguishes as three different pathways, whereas KEGG puts them all together. So what I'm actually suggesting using it as your evaluation metric is problematic because they don't contain biological pathways.
Well, it's looking at the functional coherence of genes, which I think is a different role. So, even though there are a number of functions that can be used to describe the KEGG pathway, genes are act somewhat coherently because they're involved in some sort of process or a couple of that are. The idea is that they should be a decent example of a functionally coherent set. The best example, but they are functionally coherent according to metric.
You might know something that really corresponds to the same kinds of pathways that go. And at one point, which is, I found your graphs really hard to interpret because any term we're actually getting pathway in your the viewer.
We can talk afterwards.
Hi. If I understand correctly, this being information content is working rational by choosing this and not
what?
resting measure or objectively look at their information because just looking at absolute different tell if they're not as G's. It's just
we have as part of the package a weighting metric that we used very easily to different metrics and compare which metrics are most affirmed of the term relationships. So
it was just behind.
I'll know that there's a number of metrics out there.
Oh.